Hi students, this is A. Mandri Kumar Rajesh. In this video, we are going to discuss a topic called Salient features of Ascomycetes. Ascomycetes are otherwise called as sac fungi. I give you the reason why they are called as sac fungi within a few minutes. Let us talk about the salient features of Ascomycetes. Ascomycetes include a wide range of fungi such as yeast, powdery mildew, morels, Penicillium, Aspergillus, Polystigma, Morchella and so on. So the class Ascomycetes includes wide variety of fungi. Majority of the species live in terrestrial environment. So most of the fungi which belongs to Ascomycetes can live in terrestrial environment. They are land dwellers. But some of the fungi can live in aquatic environment. So that fungi can live in freshwater and marine water ecosystem. The third point, the mycelium is well developed, branched with simple septum. Actually, Ascomycetes comes under higher fungi. So the mycelium is well developed, branched with simple septum. It possesses the cross wall. In unicellular forms, the cell wall is made up of glucans and mannans. So in unicellular fungi, for example, yeast is an example of unicellular fungi. So the yeast cell wall is made up of glucans and mannans, whereas in septate fungi, it consists of chitin and glucans. So in the septate fungi, the cell wall is made up of chitin and glucans. Majority of them are saprophytes, but few of them are parasites. For example, Erypsae is a fungi which can cause the disease powdery mildew. So the fungi which belongs to the class Ascomycetes can cause disease to the plants and animals. Then some of the fungi can live in or grow on the cow dung. So that fungi are called as caprophilous fungi. Example, Pachysa. Pachysa is otherwise called as cup fungi. The sixth point, asexual reproduction takes place by fission, budding, idea, gonidia and chlamydospore. So fission means a single cell can divide into two equal daughter cells, and that condition is called as fission. Budding means a soft small portion of the cell wall bulges out and the daughter nuclei migrated into that bulges. Then the bud pinches off by the process constriction and that bud develop into a new fungi, and that condition is called as budding. Idea means it is otherwise called as thalospore, orthospore. The cell of the hyphae at the distal end get round off and separated from that fungi. Then that separated part can produce or able to develop into a new fungi, and that spores are called as idea or thalospore or orthospore. Gonidia means that spores are produced externally on the tip of the specialized hyphae. So gonidia means that spores are produced externally on the tip of specialized hyphae, and that spores are called as gonidia. Then chlamydospores. So chlamydospores are otherwise called as resting spores. During unfavorable condition, they are in dormant condition. The chlamydospores are formed by the formation of thick wall around the cell. Then this chlamydospore, during favorable condition, this chlamydospore are able to develop into a new fungi. So that's all about the asexual reproduction. Then I am going to tell about the sexual reproduction, how the sexual reproduction takes place in the Ascomycetes. Then how the ascospores are produced inside the ascus or ascogenous hyphae. Let us talk about sexual reproduction in Ascomycetes. Sexual reproduction takes place by the fusion of two compatible nuclei. Now I am going to explain the sexual reproduction process in Ascomycetes step by step. So please listen carefully. The first point, plasmogamy is not immediately followed by karyogamy. First you have to understand what is called plasmogamy and karyogamy. Plasmogamy means the fusion of protoplast, two parent protoplasts without nuclei, and that condition is called as plasmogamy. Karyogamy means fusion of nuclei, and that condition is called as karyogamy. So here plasmogamy is not immediately followed by karyogamy. Only fusion of protoplast takes place, not fusion of nuclei takes place. That is why after plasmogamy, antheridial, that is the male sex organ, and ascogonial, that is the female sex organ, nuclei are pair up within ascogonium, resulting in the formation of dikaryon. So here you can find two nuclei, one from antheridia, another from ascogonium. So here you can find two nucleus, and that condition is called as dikaryon, and that condition is called as dikaryon. Karyon means nuclei, di means two, so it contains two nuclei. Ascogonium contains two nuclei. The dikaryons passes into ascogenous hyphae. So the dikaryon passes into the ascogenous hyphae. The tip of the ascogenous hypha bends, forming hook, and is known as groicer. So here you can find a hook-like structure and is called as groicer, and it is called as groicer. So here you can find a hook-like structure. So the tip of the ascogenous hypha bend, forming a hook, and is known as groicer. Then the nuclei of groicer divided by conjugate division. So the nuclei of the groicer divided by conjugate division. So here you can find four nuclei. Here you can find four nuclei. So that nuclei are divided by conjugate division, and they are parallel to each other, So in the first step you can find dikaryon. In the second step, the nuclei are divided by conjugate division. So here you can find four nuclei. Then during the division of the nuclei, two septa appear between the two daughter nuclei. So here you can find two septa, two cross wall during that division. So here you can find the cross wall, and here also you can find the cross wall. So during the division of the nuclei, two septa appear between the two daughter nuclei. So here you can find the cross wall. Basal cell. So due to that, the formation of cross wall, they are formed into three different cells. Due to the formation of septa. They are basal cell or stall, penultimate cell or dome cell, then ultimate or tip cell. So this cell is called as basal cell or stall cell. So this cell is called as basal cell or stall cell, and this cell is called as penultimate cell. This cell is called as penultimate cell, and this cell is called as ultimate cell or tip cell. This cell is called as ultimate cell or tip cell. So these three types of cells are produced by the septa. So during division, the septa are formed. So due to the formation of septa, three types of cells are formed. They are basal cell, penultimate cell, tip cell or ultimate cell. Then I am going to tell you the remaining points. The next step, the stalk cell and the tip cells are uninucleate, whereas the penultimate cell is binucleate. So here you can find the basal cell which possesses only one nucleus, and the tip cell, here you can find the tip cell and which possesses only one nucleus, while in the case of penultimate cell it possesses two nucleus. That is why they have mentioned penultimate cell is binucleate. Then the penultimate cell is the ascus mother cell. So this penultimate cell is considered as ascus mother cell because it contains two nuclei. two nuclei. Then the two nuclei fuse to form a diploid nucleus in the ascus mother cell, which elongates into an ascus. So these two nuclei get fused, results in the formation of the diploid nuclei. Results in the formation of diploid nuclei. So these two nucleus get fused, results in the formation of diploid nuclei in the ascus mother cell, which elongates into an ascus. So this ascus mother cell get elongate into an ascus, So it get elongated. Then the diploid nucleus undergoes meiotic division to produce 4 haploid nuclei. So this diploid nucleus undergoes the reduction division. Due to the meiotic division, the diploid nucleus get reduced into 4 haploid nuclei. Then, which is further divided mitotically to form 8 nuclei. So this 4 haploid nuclei undergoes mitotic division. Due to mitotic division, the 4 haploid nuclei become 8 haploid nuclei. Then the nucleus get organized into 8 ascospores. The ascospores are found inside a bag-like structure called ascus. So this is an ascospore. So this ascospore present inside a sac-like structure. That is why this fungi is otherwise called as sac fungi. So the ascospores are present inside a sac-like structure. That is why this fungi is called as sac fungi. Then asci get surrounded by sterile hyphae forming fruit body called ascocarp. So this is called asci. So you can find number of ascus. Here you can find number of ascus. So that is why they have mentioned asci in plural form, asci. So that asci are surrounded by a sterile structure called ascocarp. So this is the ascocarp, the fruiting body which surrounds the asci, So the asci can be classified into 4 types. They are Clustothecium, Perithecium, Apothecium and Pseudothecium. Clustothecium means that ascoscarps are closed. Clustothecium means, clusto means closed. So that ascocarp are closed. So that ascospores are released when that ascocarps are degraded or decayed. So during degradation or decaying of that particular ascocarp, at that time only the release of ascospore takes place, and that ascocarp is called as Clustothecium. Perithecium means, so this is the structure of perithecium, vertical section of perithecium. So it is ascocarp look like a flask shape, round, or it looks like a flask shape. Then it contains asci. In between the asci you can find paraphysis, a hair-like structure which gives support to this asci. Then it contains osteole. So this is a pore. Through this pore only this ascospores are released. Perithecium means that ascocarp are flask or round shape, and it contains osteole, a pore. Then apothecium. So apothecium means they are open and they are cup shaped. So you can find that ascocarp are cup shaped and they are open. Example, Pessisa. The diagrams are given in our book. Pessisa is otherwise called as cup fungi because that ascocarps are cup shaped. Then pseudothecium. Pseudothecium means it resembles the perithecium ascocarp, but it possesses ascospore but not in that organized form. That is why they are called as pseudothecium. So that's all about the salient features of Ascomycetes. If you have any doubt, please clarify with me. Thank you very much for watching this video. Thank you.